Welcome, everyone. Thank you for joining us for today's keynote presentation, A Comparative Approach to Oncology Drug Development, Integrating Canine Models, presented by Amy LeBlanc, Director, Comparative Oncology Program, Center for Cancer Research, National Cancer Institute, National Institutes of Health. My name is Xavier Gutierrez, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. LeBlanc. I will now turn the presentation over to her.
Thank you very much, Xavier, and a big thank you to the organizers of the event and to LabRoots for facilitating this presentation. I'm thrilled to have this opportunity to share with you our work at the National Cancer Institute and within the Center for Cancer Research, which is the Intramural program of the NCI, and tell you about how we integrate dogs that naturally develop cancer into studies of cancer biology and cancer drug development. So the first concept that we'd like to talk about in comparative oncology is how similar are dogs and people to each other. And this sort of advertising slide tells you and sort of gets across the point of how we may look similar on the surface, but also more importantly, a lot of the diseases that affect dogs and people are the same. And more we understand and appreciate differences between adult humans and animals and the distinguishing factors of pediatric diseases and those that affect our companion pets that live in our homes. And so when we talk about comparative oncology, the distinction becomes very specific to the types of cancers that dogs and people may get and may have in common with each other. And indeed, there are remarkable similarities in cancers that are shared between humans and dogs. For the purposes of today's presentation, we'll be focused primarily on the companion pet dog. And in this particular publication in 2015, it really captures a lot of the depth and breadth of types of cancers that are shared between dogs and humans. And in that sense, it becomes a really important concept to understand these diseases, not just for the benefit of dogs, but also for the people who love them and care for them. And in doing so, we learn a lot about the types of cancers that affect us and can leverage this information to help both species. So the National Cancer Institute created the Comparative Oncology Program to help really dig into this question about the value of a comparative and integrated approach to cancer drug development. And in doing so, we strive very much to understand and appreciate these naturally occurring spontaneous cancers that develop naturally in dogs. And they really do represent a huge spread of different types of tumors. So solid tumors, both epithelial and mesenchymal in origin, but also the hematologic cancers. And these cancers develop in dogs that are an immune competent host. And in that sense, their tumors co-evolve with the stroma around them. They are not experimentally induced or xenografts in any way. And they also develop in dogs that unfortunately have a fairly compressed disease progression and a short survival time. So that really does, though, turn into an advantage when studying these diseases because you have a reasonable amount of time to investigate things like treatment strategies and different diagnostic methods that really help us understand how the natural progression of the cancer can be appreciated in a reasonable amount of time. And then also within the scope of veterinary medicine over the last 30 years or so, veterinary oncologists have really become quite expert in applying chemotherapy, surgery, and radiotherapy techniques, both alone and in combination with each other, mirroring how physician-based medical oncology and radiation therapy and surgical oncology are applied for human patients. And we mimic a lot of our approaches on how things are done for human patients. And that becomes a translationally valuable approach that then helps us work with our physician colleagues to understand how studies can be carried out in dogs that mirror what happens on the physician side. And then also dogs that develop cancer also naturally develop therapy resistance and naturally occurring metastasis. And so those types of phenomena are not often or always reflected in experimental mouse models of cancer. And so they become very valuable facets of the types of diseases that we see. And sort of the last point on this slide is that there's no true standard of care in many cases for dogs that are presenting for care for their cancers. We don't have insurance reimbursement that's driving the choices that people can and can't make. And so ultimately, the pet owner has a lot of flexibility in determining what is the best fit for their pet and for their family when choosing to pursue a treatment strategy versus just palliative care for their pet. So this figure really helps to drive home where all of these facets really help us position the dog within the sort of standard drug development paradigm, where preclinical models like mice and laboratory beagle dogs and non-human primates are heavily studied early on for the purposes of safety and tolerability of new drugs. The tumor-bearing pet dog can help inform as that path moves forward into human studies in a variety of ways. So studies that are carried out in dogs can help answer questions about a drug's pharmacokinetic and pharmacodynamic profile in a way that may be impossible to observe longitudinally in a mouse model. We can also investigate combination strategies and select very specific responding histologies and dive more deeply into comparative studies of dose and schedule after the recommended after the phase 1 work is conducted in people and we're heading into a recommended phase 2 dose for human studies. And finally, you can think about ways that dogs can participate in post-approval setting where we're looking at potentially evaluating a drug in a new tumor type or a new indication. So there's value to be had, I think, at many different steps during the classic drug development path. So the idea of comparative oncology was recently highlighted during a National Academy of Medicine workshop. And if you're interested in more information, you can read sort of the post-workshop impressions of the folks that participated and responded and gave us feedback during this workshop. So this paper was published shortly after the workshop was conducted in 2016 and really served to highlight the opportunities in comparative oncology moving forward. Maybe most importantly, this manuscript serves to offer the challenges that we face and the opportunities that we have to address to really drive home that the integrated approach does have value and can optimize the way that we develop drugs for human use, but also helps us shed light on the types of cancers that are affecting the dogs that we share our lives with and how they can benefit from this work as well. So the comparative oncology program that I direct sits within the Intramural research program of the NIH. And so this slide just sort of shows you where we sit organizationally within the entity of the National Cancer Institute. Our parent branch is in molecular imaging. And I've outlined the DCTD, which is an extramural division, the Division of Cancer Therapy and Diagnosis, because we work closely with them on a number of clinical trial efforts and drug development programs. So that just gives you an idea of sort of organizationally where we sit within the NCI. And one of the most important parts of our program that's worth coming into some detail about is that we have a comparative oncology trials consortium. So this network is made up of 24 member institutions, veterinary schools within the US and Canada that have signed a memorandum of understanding and have agreed to conduct and partner with us on clinical trial activities that are carried out in pet dogs with cancer and that are designed to advance questions on for human drug development. And so this clinical trial network allows us to have access to canine patients, even though programmatically we are situated within the NCI. We don't have ready access to veterinary patients on the NIH campus. So working with our clinical trial partners, we're able to deploy multi-center clinical trials across a whole spectrum of novel agents. So small molecules, imaging agents, biologic agents, devices, et cetera. And also important to note here that the consortium members also work with us to provide advocacy and access to reagents and resources that really help enable the studies to be carried out in dogs. So when you work with dogs, there's an obvious need for canine-specific reagents and assay expertise, cell lines, tumor microarrays, antibodies, et cetera. And so having a network of experts within the veterinary academic world allows us to really connect trial sponsors with the appropriate pharmacodynamic assay expertise to help interpret the data and dive more deeply into questions of cancer biology. So this slide gives you an overview of all of the Comparative Oncology Trials Consortium trials that have been carried out since the program was initiated back in 2003. So you can see the status of the study, whether it's gone to publication or whether it's complete and under analysis or whether it's an open trial. And so this just gives you an idea, again, of the variety of the types of studies that we do and how we're not, sort of held fast to any one type of cancer. We certainly have an emphasis in osteosarcoma, which I will get to here in a little bit. But we really, I think, serve to connect drug trial sponsors with the appropriate patient population and really spend a lot of time digging into the types of questions that the dog could, within the context of a clinical trial, we can ask and answer questions that may not be possible or feasible to ask in other species, and that would be critical to a drug's development prior to or in line parallel with human use. So, and then this slide just sort of gives you the idea of the scope of the types of biologic samples that can be collected from dogs with cancer. So we have many, many opportunities to collect a whole constellation, really, of sample types, not just tumor and maybe matched normal adjacent tissue, but also a full complement of biologic fluids like serum, whole blood, plasma, urine, et cetera, that are really helpful for post hoc analyses and for biomarker mining and things like that. And also remember that the dog's physical size and their clinical observation within the care of their tumor and their enrollment on a clinical trial allows us to serially sample these types of tissues and fluids that really enable some in-depth questions to be asked during the time of drug exposure. So a really valuable opportunity there lies within each patient that is enrolled on these trials. So next I want to tell you a couple of stories, examples of specific trials that our program has run. And the first one is a study that we did in naturally occurring canine melanoma and an interleukin-12-based immunocytokine therapy. So canine melanoma and human melanoma have some very interesting similarities and differences. And so visually, when you look at these tumors, they look fairly similar. They're often pigmented. And in the case of the dog, the most common location is actually within the oral mucosa, the lips, tongue, hard palate, and gums, sometimes also around the lip margin. But in humans, typically melanoma is a dermal disease in the skin and often linked to UV exposure. But there are the rare cases of human oral mucosal melanoma depicted here. What's also interesting is that when you dive more deeply into the biology of melanoma in people and in dogs, they have some things that are in common that are quite interesting. They have shared pathway activation within the AKT and MAP kinase pathways and seem to exhibit similar loss of P16 and P53 among other tumor suppressor genes, but they also are similarly aggressive biologically, and they exhibit prompt recurrence after resection if incomplete and have a wide propensity for metastases. And they're also very poorly responsive to standard small molecule cytotoxic chemotherapy and seem somewhat responsive to immunotherapy. So those similarities and differences really have led the community to believe pretty strongly that canine melanoma is a nice model for human melanoma, and therein lies a lot of opportunity for immunotherapy-based drug development. So to that end, we conducted a trial of an interleukin-12 immunocytokine, so IL-12, fully human, linked to a IgG based immunocytokine that had a homing peptide that recognizes necrosis. And this agent, NHS-IL-12, was evaluated in a solid tumor study of dogs and had an expansion cohort in malignant melanoma. And depicted on the slide is one of the biggest responders. So this is a dog that had a really bulky maxillary melanoma that had a complete response to this drug. Visually, you can see that in these photographs. And this trial was really important because it was a fully human construct. And so, we were not sure necessarily how many repeated doses of this agent could be given to dogs, given that this was not a dog-specific immunocytokine. But what I'll show you are some of the data that really helped move this agent forward and sort of give you then the recent update of where this agent stands in its human development path and why the dog data was so important to that progress. So first of all, I just wanted to show you that within this trial, this is an example, and in this trial, this is the actual study schedule and the procedures that were carried out during the trial. And so you get appreciation here for the clinical management of the dogs that were enrolled on this trial, where we have eligibility and baseline assessments, baseline tumor biopsy, and draining lymph node aspiration cytology and flow cytometric assessment. We did clinical staging and photographed all of the target lesions we were monitoring, and then during drug exposure, we're able to serially sample serum, PBMCs, and standard biochemical and hematologic monitoring to really capture both safety and tolerability, and then the pharmacokinetics and the pharmacodynamic response to the drug over about a 4-week period. And the dogs that had were responders to the agent were allowed to move forward onto additional treatment cycles every 28 days. And so what we were able to learn from this is that dogs that receive NHS-IL-12 subcutaneously achieve measurable drug exposures, and this was a dose escalation trial, so there were multiple dosing cohorts that spanned from 0.4 mg per meter squared all the way up to 2.4 mg per meter squared. So in this slide, I'm showing you that after the time of administration of drug, we were able to capture drug levels in serum over several hours post-administration across all the dosing cohorts to just define the PK profile. And then similarly, dogs that receive the drug subcutaneously demonstrate the expected pharmacodynamic or PD response post-treatment, in that they have measurable release of interferon gamma and IL-10 that again could be captured serially in the hours after drug administration occurred. And here you see again an example from each of the dosing cohorts. And then here's the IL-10 data as well, similarly showing the spike in IL-10 release after drug is administered. So the deliverables from this trial were many, and I think I spend a few minutes kind of highlighting this, that this is a fully human necrosis-targeted IgG1 antibody IL-12 conjugate. We found it can be safely and repeatedly administered to tumor-bearing dogs and does elicit the expected PD biomarkers. We also, because we had access to bulky disease, were able to serially biopsy the lesions undergoing treatment and look at things like CD8 positive tumor-infiltrating lymphocytes and other changes in histologic appearance, and try to link all of those PD biomarkers to exposures that were captured with the pharmacokinetic assessment. And within the cohort of these dogs, we identified an efficacy signal in 2 of 7 dogs with bulky oral malignant melanoma. We were also similarly able to capture the adverse events that these dogs underwent and exhibited, and all of those were relatable to the expected cytokine release that you would see with exposure to IL-12. So a self-limiting lymphopenia, dogs developed fevers that were self-limiting. They did have hepatic enzymopathy, again, that you would expect to see. When we got into the higher dosing cohorts, and that's above 1.6 mg per meter squared, we did see some serious grade 4 and 5 events that indicated to us that the MTD was probably lower within our dosing cohorts, and we were able to expand the cohort at the 0.8 mg per meter squared dose level and really nail down the maximum tolerated dose in the dog, the tumor-bearing dog, to be 25.8 micrograms per kilo. And it's important to recognize, too, that these are patients that are pet dogs. And so these are not healthy 2-year-old beagles. These are 10, 11, 12-year-old dogs that, even though we have very strict eligibility criteria for hematologic and biochemical parameters to be eligible for the trial, it's possible that they have some silent hepatic or renal insufficiency that doesn't exhibit itself clinically. But in that sense, they resemble a pretty typical phase 1 human patient. So they're not a young, healthy 18-year-old. They're, more representative of a geriatric population. So also, I think pretty important to really making the case here is that the company that brought forward this agent for study in dogs had received the dog data and then chose to reprioritize the development of this agent. And so they opened an IND for this agent in 2011. The dog data was collected over about a 3-year period, 2007, 2008, 2009. And then the IND for human use was opened at the clinical center here at NIH in 2011. And that data from the phase 1 was recently published in Clinical Cancer Research. Within that phase 1 trial, they had enrolled 59 patients and were able to identify the maximum tolerated dose as 16.8 micrograms per kg, and that occurred at dose level 8. And just as a reminder, we identified the MTD in dogs to be 25 micrograms per kg at dose level 2. And so very similar MTD between human and dog. And in fact, the dog might have a slightly higher MTD. Hard to say with the number of patients that we enrolled, but I would definitely say that at dose level 2, this was an extremely well-tolerated drug in the dog. And we were able to capture an adverse event profile that was extremely similar between humans and dogs, and also echo the pharmacodynamic data being very similar between humans and dogs. And so we generated the data, the clinical management data in the dogs, for about $300,000 in about 18 months. And unfortunately, it took a lot longer and many, many, many more patients on the human side to really arrive at similar findings. So 7 years and 59 patients compared to what we were able to identify and learn in a relatively short period of time. So I think that this story really helps to understand the trajectory of dogs and where they can really help inform and potentially help optimize or streamline some of the follow-on work in people, perhaps maybe as that relates to a starting dose or recognition of where kinetics of pharmacodynamic responses can be tailored based on what we learn in dogs. And those studies can then be a little bit more simpler and quicker and easier to recruit on the human side. So if there's interest, this is the first in human phase 1 work. This just came out last August in Clinical Cancer Research for the NHS IL-12 human trial. So next, I would like to change gears and talk a little bit about our work in canine osteosarcoma, specifically in our clinical trials that are designed to ask questions about ways that we can target metastatic progression. So canine osteosarcoma is an extremely common canine disease. It affects at least 10,000 middle-age to older, large and giant breed dogs in the United States. Some estimates maybe take this incidence up towards 100,000 or even more dogs affected every year. So extremely common and very serious health problem for older dogs here in the US and beyond. The standard of care currently is amputation of the affected limb followed by adjuvant chemotherapy. But even doing that only allows us to have long-term survival or a cure of 3 years or longer of about 5 to 10%. And unfortunately, the median survival time overall, even despite aggressive therapy, is only about 10 to 14 months. And the vast majority of dogs succumb to metastases or spread, typically into the lung, but also other places too, like the bone and soft tissue. And really, once dogs develop metastatic disease, we don't really have good, effective management strategies for that advanced stage. And so drawing some parallels between humans and dogs are obviously, important of oncology as a whole. Back in 2009, a very nice paper came out of our program under Sean Khanna and Melissa Paoloni that really sought to ask questions about how similar at the genomic or gene expression level are dogs and humans and their tumors. And this paper, the heat map there on this slide, shows you that from a gene expression standpoint, you actually cannot distinguish dog from human tumors. And compared to normal healthy bone, those are indistinguishable as well. So it really gives us a lot of opportunity to think about the clinical similarities and then have that molecular underpinning that's so similar really support ongoing studies of this disease in both species. Pediatric osteosarcoma, in contrast, is an extremely uncommon disease. So less than 1,000 children are diagnosed with this disease in the US. These children undergo neoadjuvant chemotherapy with follow-up limb salvage. Even with, again, the aggressive treatment approach, 70% of them will have long-term survival, which means 30% of them will succumb, again, mainly due to metastatic disease within 5 years of diagnosis. And there really aren't any good predictors or effective treatment strategies for metastatic disease in children either. So based on those similarities, we really have, I think, a lot of opportunity to really think about ways to study metastatic progression and metastasis biology in the dog that presents with osteosarcoma and learn translationally where our targets and opportunities could be, and then relay that to the pediatric oncology community. So lots of studies and literature exist in this space to help back up the predictability of fatal metastatic disease within 1 year of diagnosis. And so these are just 6 studies of many that really nail down the reality of how hard this disease is to treat in the dog, and how there's a real opportunity for us to think about strategies that could enable a prolongation of progression-free survival. And if we could significantly extend progression-free survival in dogs with osteosarcoma, then we could really feel confident about that being an opportunity for study on the pediatric side. And so one of the studies that I'll tell you about today is a trial that's headed up by our colleague and friend, Nicola Mason at the University of Pennsylvania. She is studying a HER2 targeted listeria immunotherapy and has published some really promising phase 1 results in clinical cancer research. And so this observation that dogs that receive this listeria-based immunotherapy, in addition to having standard of care, live significantly longer than a control group of historically controlled standard of care therapy patients. So we sought, with the help of the Morris Animal Foundation, to extend upon these observations and really nail down whether or not listeria-based immunotherapy could have a future, not just for dogs, but also for pediatric patients. So partnering with the Morris Animal Foundation's Osteosarcoma Project has allowed us to leverage our clinical trial consortium membership through a longstanding initiative and fundraising efforts to understand and gain access to a model for assessing new treatment strategies that are really focused on targeting metastatic progression to benefit both dogs and humans. And so the concept here is to study a series of novel agents from a variety of sources through canine trials that all run through our COTC mechanism, and compare those novel agents against a contemporaneously accruing standard of care control arm. And that allows us to have a fully randomized prospective design with a one-to-one enrollment between control and investigational drug. And these studies are powered to detect a 50% improvement in disease-free interval over standard of care. And that is really an important point, because we need to be able to show a significant amount of improvement in disease-free interval or progression-free survival to really have that be meaningful for adoption on the pediatric side. And to that end, actually, this paper published in Clinical Cancer Research in 2014 reflects an algorithm for agent prioritization and how this group of key opinion leaders came to consensus on metrics for valuing agents through a variety of different data types. And this paper talks about how data that's collected in tumor-bearing osteosarcoma patients can be included in evaluation scheme that helps the pediatric side understand and rank agents that could be considered for further study. So the Comparative Oncology program has taken this on as an initiative that we spend a significant amount of time and resources towards conducting these trials and making sure that they are implemented with the guidance of our comparative oncology colleagues within both the veterinary and pediatric oncology communities. This is the summary of current Morris Animal Foundation sponsored Osteosarcoma Project clinical trials. So at the top, you see COTC 022, which is our standard of care arm. Standard of care in this schema is defined as amputation of the affected limb, followed by 4 doses of adjuvant carboplatin chemotherapy given at 3-week intervals. And then the investigational arms currently under study, there's 2 of them. There's COTC 021 and COTC 026. 21 trial is looking at adjuvant rapamycin as an inhibitor of mTOR. Again, that's added to standard of care. So every dog on these trials receives the standard of care therapy, and they're randomized to either receive adjuvant rapamycin or standard of care. And then our adjuvant listeria trial that's currently open, those dogs are being enrolled again to provide the statistical power compared to the 022 trial. And so all those listeria patients are receiving the listeria, again, in the adjuvant form, so after they've completed standard of care. So we opened the first 2 arms of the trial back in the fall of 2015, and we're able to recruit over 300 dogs in about 2.5 years, or just shy of 3 years. And then we have had the 026 trial open since the fall of 2017. And we've got 91 at current count out of 110 for the target enrollment. So I'd say, we have a lot of success in enrolling large numbers of dogs. Again, I think that reflects the fact that this is a funded trial mechanism, and pet owners really appreciate the opportunity to have care carried out through this mechanism, and that this is just a really common disease as well. So it really does serve the needs of both the veterinary and the pediatric communities to have these types of trials open. And this is the study schema for the standard of care arm. So dogs are screened and confirmed as eligible, and they undergo surgery to amputate their affected limb. And we bank the tumor and all of the pretreatment samples from serum and PBMCs, et cetera. And they are then clinically followed with thoracic radiographs and physical exams every 8 weeks after they complete carboplatin chemotherapy. And so this is just a snapshot of the types of dogs that have been enrolled and recruited to the standard of care arms. We started out with 165 that were randomized. And then after the process of confirming the disease and confirming that they are free of metastasis, we have 149 dogs that have survived the postoperative period and started carboplatin chemotherapy. And this cohort currently, this is not fully mature data, but it's pretty close to being complete. We'll have a fully mature data set in the fall of 2019. So in about 8 more months, we'll have this all complete and ready to share with the community. Currently, this data set shows us that the median disease-free interval of the intent to treat population is about 5.8, just shy of 6 months. And that's in line with what previous reports have shown. It might be a little bit shorter than some of the statistical comparisons that are out there in the other osteosarcoma published literature. I think that reflects the fact that we had very prescriptive follow-up and very careful evaluation of thoracic radiographs, both during and after carboplatin chemotherapy. So some of that observational bias may be could serve to explain why our disease-free interval is a bit shorter than what's been published by other investigators. But I think the key point here is that this standard of care arm and all of the other Osteosarcoma Project initiatives and clinical trials really help provide a strong, scientifically sound backbone for which comparison of novel agents can be assessed, particularly in the minimal residual disease setting, where we know it's so critical to address metastasis before it becomes clinically evident, because once metastatic disease sets in and is bulky, the treatment options that are effective become extremely limited, if not non-existent. And that these adjuvant trials that we're currently working on, the 021 and 026 trials, really serve as nice proving grounds for these agents to be considered for pediatric assessment. And we have really, we strive always to engage the stakeholders within drug development, but also patient advocacy groups, and specifically the Children's Oncology Group is a major stakeholder in our activities. So we're really, it's really important to us to make sure that whatever we do has translational benefit for both dogs and for humans. And to that end, actually, Advaxis, the company who has created and manufactured the Listeria construct, has now entered into an agreement to have OS therapies move that agent forward for human clinical trials. And so that relationship between Advaxis and OS therapies is pretty critical. And I think that the data that we're generating in dogs is really helping to really garner the enthusiasm for the movement of this agent forward. And there's a parallel product that will be produced and licensed and moved out into veterinary use from Aratana. So this is an example of where the work that we do really has bidirectional benefit for both children and for dogs. I just wanted to end with a couple of coming soon points. So the National Cancer Institute's Human Genomics Data Commons, or the GDC, is a portal for which we are planning a comparative initiative in dogs. So we are creating a canine genomic data commons. It is going to be much more really than just genomics, though. The idea here is to have the opportunity to provide the cancer research community with a publicly accessible data repository of dog cancer data that will help investigators really have the tools and the opportunity to query what we know about dog cancer in context of human cancer, and allows opportunities for people to put their data that they generate in their labs or with their trials and studies into one place where we can then curate a really high quality integrated data set from dogs that will again help drive home the value of studying cancers in dogs and really open up new opportunities to have clinical trials and other activities carried out in dogs that are informed by the integration of the genomic data between dogs and from people. So we have an exciting time, I think, ahead of us in comparative oncology. We think it's really important to help continue to emphasize a scientific dialogue about the value of the dog and the dog that develops cancer naturally within its lifespan, and how learning more about those patients can really help understand where they can be strategically placed in clinical trial studies and other scientifically robust assessments of biology and drug development. So, you know, we certainly don't believe or advocate that the dog will solve all the problems with cancer drug research and development, but we think that if there's careful consideration of where the dog can really inform and complement the existing drug development activities that involve other species and involve humans, we can get to better drugs faster and maybe take less promising drugs off the list sooner. It's also, I think, really important for us to maintain the highest standards we can for clinical trial design and execution and reporting. And so to that end, we spend a lot of time mirroring and understanding how clinical trials for humans are put together and really adopt those best practices for our work as well. And I think finally, it's a community-based initiative, and this came through earlier in my talk when I was talking about the National Academy's workshop on comparative oncology. One of the biggest gaps really in our knowledge has been getting a more robust assessment and understanding of canine-specific data sets and canine-specific reagents and correlative assays in place to help enhance those activities and really help improve the translation to humans. So that is a challenge, I think, for us as a community to continue to develop those data and those resources and the necessary tools to help enable the scientific discovery to continue and be relatable to humans. So with that, I'll conclude my presentation and send a huge thank you to, again, LabRoots and the organizers of this event, and to all of my colleagues within the NCI and the Comparative Oncology program, and all of the various partners, collaborators, and champions that we've had both within NCI, NIH, and within the drug development community as a whole. And a special thank you to our COTC member institutions and all of the support staff, the families of the dog patients that enroll on our trials, and for everyone's hard work to help realize where the value of our study of naturally occurring cancers in dogs can really benefit both us and them. So I'll thank you for your attention and be happy to follow up with any questions via email after the presentation concludes. Thank you.
Thank you, Dr. LeBlanc, for that informative presentation. I would also like to thank Labbers for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. And as a final reminder, once again, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.